Selenium is a naturally occurring element composing up to 40% of the entire Earth's crust. It is concentrated in carbonate rock, volcanic soils, and sedimentary soils, and often associated with sulfur containing minerals and fossil fuel sources. In a natural cycle, selenium moves from rocks to sediments in soils and water. Selenium concentrations vary globally, with heavy concentrations in irrigated agricultural lands of the Midwest United States, as selenium is more abundant in groundwater below agricultural lands than it is in surface water. This results in accumulation of selenium in soils. As a trace element, selenium is essential to cell function in humans and animals. The range between deficiency and toxicity is extremely narrow and varies on an individual basis with size, genetics, and diet. Anthropogenic sources of selenium stem from mining practices. During the process of mining copper and silver, selenium is produced as a byproduct. This selenium can create concerns of contamination if there is a leak or spill that allows contact with a water source. Phytoremediation is a remediation technique utilizing plants to stabilize or remove a contaminant of concern from soil at a hazardous site. Some plant species are ideal for this technique and have the ability to extract heavy metals from the soil and accumulate high levels of biomass. Mycoremediation is another bioremediation practice that uses fungi to remediate a contaminated soil. It is a similar process to phytoremediation, and fungi are applied at the site to transform the contaminant of concern in the soil to a less toxic form, or to a state that makes it more available for extraction. The goal of our experiment is to combine the techniques of phytoremediation and mycoremediation to increase the rate of selenium removal from a contaminated soil. Both techniques are low cost and have been proven to be effective in past studies, so our intent is to synthesize these into a process that is cheap, fast, and good. Sunflowers were used in this experiment because they germinate quickly and are known to be hyperaccumulators, or a plant that is able to grow in toxic soils, taking up metals via roots and concentrating the metal in its tissues. We applied an arbuscular mycorrhizal fungi that has the potential to colonize the roots of the sunflowers. The fungi form a nutrient exchange site called an arbuscule on the roots of the plants, where the nutrients are exchanged to the host plant from the fungus in exchange for carbon. Germination rates were highest in treatments without the AM inoculant. In the treatment with the inoculant, there was a trend for higher germination rates in soil with higher contamination. The average height of the plants did not vary significantly across the treatments. The general trend showed that for plants without inoculant, tallest plants were in the 10 parts per million concentrations, and for plants with the inoculant, plants in the control group with no selenium concentration were the tallest. The results of root-to-shoot ratios across treatments and between contaminant levels were not significantly different from each other. However, there was a general trend for plants in the 10 part per million selenium soils to have a larger root-to-shoot ratio than the control or the 30 parts per million plants, which were almost the same. Elemental concentrations of leaf tissue after 4 weeks of growth indicated less than 3 milligrams of selenium per kilogram of wet plant tissue across all treatments. Cheap, low cost, maintenance, and application of sunflowers and fungal inoculant. Fast? Yep, fast germination and growth of sunflower plants. Good? Nope, our treatment failed. It didn't clean up the soil, but it didn't make it worse either, that we're aware of. We didn't see any of the trends we expected, and it's difficult to explain the trends we observed.